Hello everyone, this video is about Hematology 2 where we will be discussing abnormalities about the blood cells. The blood cells to be discussed in this set of videos would be the red blood cells, white blood cells, and platelets. In this video, we will concentrate on the different red blood cell anomalies. Let's now start an overview video about the red blood cell abnormalities and disorders. The red blood cell anomalies may be divided into different categories. We have anisocytosis, poikilocytosis, anisochromasia, abnormal inclusion bodies, and miscellaneous category. Anisocytosis refers to the increased number of red blood cells with varying size, while poikilocytosis refers to the increased number of red blood cells with variation in shape. Poikilocytosis has shape variation and anisocytosis has size variation. Anisochromasia, on the other hand, refers to the variation of pigmentation or coloration of the red blood cell. Abnormal inclusion bodies would mean that there is the presence of stainable substances or materials. Let's start with anisocytosis. Again, anisocytosis is an increased number of red blood cells, and these increased red blood cells will also have a variation in size. When we say anisocytosis, they can be divided into four different descriptions. We have the normocytes, also known as the discocytes. So these are the normal size of red blood cells, microcytes, macrocytes, and megalocyte. The first one is normocyte, also known as discocyte. A normocyte or a discocyte is a normal-sized erythrocyte. It is approximately the size of the nucleus of a lymphocyte. This is a lymphocyte, and this is the nucleus of a lymphocyte. It is said that a normal RBC, as seen under the microscope, would be the same size as this nucleus. If we see the red blood cell beside it, it is approximately the same size as the nucleus of a small lymphocyte. Therefore, this red blood cell is considered as a normocyte. The size of a normal red blood cell is 6 to 8 microns, or 80 to 100 femtoliters. An increased number of normocytes is called normocytosis, and normocytes may also be seen in hemorrhages or hemodilution. The second one is a microcyte. A microcyte is a red blood cell that measures less than 6 microns or less than 80 femtoliters. The condition with an increased number of microcytes is called microcytosis. Microcytes lack the necessary basic materials needed to develop a cell, and these are usually seen in IDA or iron deficiency anemia. In this picture, this is considered as a microcyte because it is smaller than the nucleus of a lymphocyte. The third cell is a macrocyte. A macrocyte is a big red blood cell with more than 100 femtoliters, measuring to about more than 8 to 12 microns. Seeing macrocytes in newborns is normal. An increased number of macrocytes is termed as macrocytosis, and this is usually seen in conditions with vitamin B12 or folate deficiency, so these are the megaloblastic anemias, or in situations where there is accelerated erythropoiesis. So in this picture, as reference again, is the nucleus of a lymphocyte. These are macrocytes. The last type of cell under anisocytosis is a megalocyte. A megalocyte is considered to be more than 12 microns, so these are bigger than macrocytes. The presence of megalocytes in the circulation is termed as megalocytosis and usually seen in megaloblastic anemia. So what do you call the term used when there is two distinct populations of red blood cells in the peripheral blood smear? Like for this one, we have small RBCs or microcytes, we have normocytes, and we also have macrocytes. When there is more distinct populations, what do we call this? And these are hypochromic cells and normochromic cells and hyperchromic cells. We call this as a dimorphic population. The next category is poikilocytosis. A poikilocyte is what you call an abnormally shaped cell. And in poikilocytosis, there is an increased number of cells with varying shape or an increased number of poikilocytes. In poikilocytosis, it can also be seen in a variety of pathologic conditions, meaning one poikilocyte can be seen in different types of conditions. Let's start. These are all the different types of poikilocytes, and we will briefly discuss all of them. First one is the acanthocyte, also known as the thorn cell, or the spur cell, or the stellar cell, or the astrocyte. All of these names mean the same thing, and all of them refer to this type of red blood cells. These acanthocytes are rounded RBCs with 5 to 10 spicules. These are the spicules. These are the projections that are long and irregular. They are irreversibly thorny, meaning they refer to permanent cell damage. Acanthocytes cannot return back into normocytes. This is usually seen in disorders of lipid metabolism or in severe liver disease. This is a spur. These are usually seen behind cowboy boots to use to make the horse go faster. And that is why an acanthocyte is also named as a spur cell. Now, they usually have a star shape. That's why they are also known as a stellar cell or an astrocyte. The second poikilocyte is an echinocyte, which is also known as a Burr cell. This is an example of a Burr cell with irregular sides and knobby projections. Their spines is shorter compared to an acanthocyte. And the difference of a Burr cell with an acanthocyte is that Burr cells are reversible, so they can return back into normal cells if conditions return to normal. Echinocytes may be pathologically seen in uremia. This picture is an echinocyte, which is a sea animal, and this is where this type of cell is likened into. The third poikilocyte are crenated cells. These are crenated cells looking like Burr cells. The difference is crenated cells have 10 to 30 short, blunt spicules that are evenly distributed on the surface. Burr cells have irregular spicules. Crenated cells are not clinically important. They may be due to improper smear preparation. Therefore, these are artifacts that are produced during smear preparation. The next poikilocyte is an elliptocyte or an ovalocyte. This is an elliptocyte, which is longer, and this is an ovalocyte, more of an egg shape. Elliptocytes are cigar-shaped, while ovalocytes are egg-shaped. Elliptocytes and ovalocytes are usually seen in hereditary elliptocytosis and IDA or iron deficiency anemia. The fifth poikilocyte is a schistocyte, also known as a schizocyte or an RBC fragment. These cells are disintegrating, damaged, or fragmented red blood cells as seen in this picture. We call them as schistocytes because schist means cut. These types of cells appear like red blood cells that have been cut into pieces. Therefore, there are many shapes and sizes of schistocytes. There is no specific singular description for schistocytes. As long as they look disintegrated fragments of red blood cells, they can be considered as schistocytes. This is also a schistocyte, 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 and a schistocyte. Schistocytes usually result from the passage through damaged or altered blood vessels seen in conditions like burns or DIC or disseminated intravascular coagulation. The next type of poikilocyte is a blister cell. These are two examples of blister cells where there is the presence of a vacuole inside the cell, making it look like the red blood cell has a blister. Another description is that there is a markedly thin area at the periphery. This is the area which is very thin at the side, and this is the area which is very thin at the side. Usually, blister cells are produced in trauma when the red blood cells pass through partially obstructed blood vessels. So these are damaged red blood cells that have passed through damaged blood vessels. Another example of condition where blister cells are produced is during burns. The next poikilocyte is the helmet cell, also known as the keratocyte of Behçet's, or the triangle cell, or the horn cell. These are the helmet cells. They are hollow interior resembling a helmet or a horn. So this is the one that resembles an old soldier helmet. These are caused by hemolytic processes like hemolytic anemia. The next poikilocyte is a bite cell or a degmacyte. These are examples of bite cells where it looks like the red blood cells have been bitten. This is due to the removal of Heinz bodies by the pitting action of the spleen. Let's say this is a red blood cell and there is an inclusion body, specifically Heinz bodies, present. When the red blood cell passes through the spleen, the macrophages will remove the inclusion bodies. And afterwards, the red blood cells would look like they have been bitten. This is seen in G6PD deficiency. The next poikilocyte is a pycnocyte. These are distorted, contracted, or spiculated red blood cells. They are normal in 2 to 3 month old babies, but an increase in number of pycnocytes of more than 50% of the present red blood cells is called as an infantile pycnocytosis. The next poikilocyte is a sickle cell, which is also known as a drepanocyte or a meniscocyte. So sickle cells are thin cells with elongated pointy ends. So this is an example of a sickle cell with a pointy end that resembles a crescent shape or a curved blade. Another shape of a sickle cell is the holly leaf. This is an example of a holly leaf, and sometimes red blood cells with sickle cells would look like holly leaves, like this one. So from a normal red blood cell, mild to moderate sickle cell anemia might have holly leaves appearance, and severe anemia will have the sickle cell looking red blood cells. Sickle cells may contain hemoglobin S and may be seen in different hemoglobinopathies. The next poikilocyte is a spherocyte, also known as a hyperchromic cell. Spherocytes are nearly spherical. These are spherocytes. They are smaller in diameter, and then they don't have the central pallor. Like these red blood cells, there is no central pallor present. This is present in hereditary spherocytosis or autoimmune hemolytic anemia, and they can also be produced during burns. As we have mentioned earlier, a poikilocyte may be present in a number of conditions. The next poikilocyte is a stomatocyte, also known as a mouth cell. Mouth cell is because there seems to be a mouth-shaped space in between or in the middle of the red blood cell. So these spaces are the slit-like area of central pallor. Stomatocytes are seen in hereditary stomatocytosis, and usually they indicate liver disease. The next poikilocyte is a teardrop cell, also known as a dacryocyte. These are the dacryocytes. I usually remember this term because of the word cry inside the dacryocyte, which means tear. Now, these cells are described to be tapered on one end and rounded at the other end, usually seen in pernicious anemia and thalassemia. The next poikilocyte is a Rechet cell, which is another form of a teardrop cell. So the difference is a Rechet cell is pear-shaped like a teardrop cell, but with a longer tail appearing like a tennis racket. Implications would be like the teardrop cell. Next poikilocyte has a lot of names. It is known as a target cell, a leptocyte, a platycyte, codocyte, bullseye cell, Greek helmet cell, and the Mexican hat cell. This is an example of a target cell. This is an example of a target cell. It's called a Mexican hat because it resembles a Mexican hat. In 3D, this is how they look like. So this is the red blood cell, and this is how they look like the Mexican hat. So this top would be that top. Now, it shows a central area of hemoglobin surrounded by a colorless ring and a peripheral ring of hemoglobin. So this is the central area of hemoglobin surrounded by a colorless ring and a peripheral ring of hemoglobin. Sometimes it may be a bell shape or a cup shape. That's why it has other different names. Also, it's not always pathologic because sometimes it may only be considered as an artifact. This may be seen pathologically in IDA or iron deficiency anemia and other hemoglobinopathies. The next poikilocyte is the pincer cell. This is an example of a pincer cell from the word pins. Okay, these are mushroom-shaped cells. It looks like you pinched the sides of a red blood cell and it looks like a mushroom. That's why it's also called a pincer cell. And these are treated as spherocytes. The next category is anisochromasia. Anisochromasia means that there is variations in hemoglobin content. We use the MCH and MCHC as guidelines. The different types under anisochromasia would be normochromic, hypochromic, hyperchromic, polychromic, annulocyte, and target cell. The first type of cell is a normochromic or an orthochromic cell. These cells have normal hemoglobin content, and there is a 1/3 clear central pallor. These are normochromic cells with 1/3 of the inner part of the red blood cell being pale. The next type of cell is a hypochromic cell. These have a decreased amount of hemoglobin, a decreased result for MCH and MCHC. The central pallor is large with more than 1/3. Remember, a normal one should only have 1/3 clear central pallor. Usually, these would also be microcytes seen in IDA and thalassemia. So these are examples of hypochromic cells with more or bigger central pallor, more than 1/3 of central paleness. Another type of cell is the hyperchromic cell. A hyperchromic red cell is also known as a spherocyte. Spherocytes have no central pallor. Another type of red blood cell is a polychromic cell, where it looks like slight blue-orange in color when using the right stain, seen in reticulocytosis, and this is slightly macrocytic. The next type of cell is an annulocyte or a pessary cell, also known as a ghost cell. So these are cells with a very thin rim of hemoglobin and a central large area. So this is an annulocyte looking like a ghost. That's why it's called ghost cell. You only see the very thin rim of hemoglobin. This is the annulocyte or the ghost cell. A target cell is also considered under anisochromasia. Let's now proceed to the next category, which is about inclusion bodies. These are the different types of inclusion bodies which we will discuss. The first one is the Howell-Jolly bodies. Howell-Jolly bodies are spherical and they are the nuclear remnant of a red blood cell. This is a Howell-Jolly body. There may be multiple or they may be single Howell-Jolly bodies in a red blood cell. They stain reddish blue to purple. These are seen in hemolytic anemia and pernicious anemia. The second inclusion body is a Cabot ring. These are thread, color blue. They may be ring-shaped, twisted, like the figure of 8 in the cytoplasm. They are seen in severe anemia and lead poisoning. So here is one Cabot ring. This is the figure of 8 Cabot ring. Next is basophilic stipplings. These are multiple or fine coarse RNA inclusions. The blue spots are the basophilic stipplings. These cells will not stain with Prussian blue and are present in lead poisoning. The next inclusion body is the Heinz-Ehrlich bodies. This is the Heinz body, Heinz body, Heinz body. Description is that these are inclusions from denatured hemoglobin. They are membrane bound. That is why they are at the periphery. Here it looks like it's going out. So that's the Heinz-Ehrlich body. G6PD deficiency. Okay, so in G6PD deficiency, we will find Heinz-Ehrlich bodies and stained with crystal violet and crystal blue. The next cell is the siderocytic granules or the Pappenheimer bodies. These are actually iron granules that have clustered into one end, as this picture shows. They stain dark violet. Siderocytic granules and Pappenheimer bodies are actually the same. It only depends on what stain is used. Siderocytic granules are seen when using Prussian blue stain, while Pappenheimer bodies are seen when using Wright stain. These types of inclusion bodies are usually seen in sideroblastic anemia or other hemoglobinopathies. Next inclusion bodies are malarial stipplings. These are granules that are formed by malarial parasites. Examples of these stipplings would be produced, for example, by Plasmodium vivax, which produces Schaffner's dots. Plasmodium malariae produces Siemens dots, while Falciparum produces Marur's or Christopher's dots. So these are all the dots seen on the red blood cell. Next would be the hemoglobin crystals. So these are rod-like or angular, opaque, and sometimes they are curved reddish-brown hemoglobin crystals. They are also called as the Washington Monument. For example, is this one. And they are seen in a hemoglobin C or SC disease. Next would be the hemoglobin H inclusions. So they are small greenish or blue bodies due to the instability of hemoglobin H in the supravital stain. So these are the hemoglobin H inclusions. These are seen in hemoglobin H disease. The next type of cell is a nucleated red blood cell. So these are usually orthochromic normoblasts. So these are immature red blood cells that may be seen in the peripheral blood smear. These are usually seen when there is marrow stimulation on where or when the marrow finds a need to produce more blood cells. This is normally seen in newborns. Next would be the miscellaneous RBC appearance. So we have the rouleau formation and agglutination. Rouleau formation is when the red blood cells are stacked together like a stack of coins. So this is one rouleau formation. Rouleau is not clinically significant, but you have to make sure that you do not mistake it for a real RBC clumping. Agglutination is the irregular and round clumping of red blood cells. So this is one agglutinate, and another agglutinate. Seen in cold agglutinin and autoimmune hemolytic anemia. Next, we have the different erythrocyte disorders. So first, we can differentiate the erythrocyte disorders starting with an RBC concentration. So it can either be an increase or a decrease. Increased erythrocyte disorders would be polycythemia vera, and a decrease would mean that it is anemia. Polycythemia is caused by the mutation of the JAK2 gene, specifically the V617F mutation. While in anemia, there can be a lot of different causes of the condition. It can be a bone marrow failure or a systemic disorder. It may also be due to the abnormal iron metabolism or abnormal hemoglobin development. Anemia is a condition of a decreased red blood cell condition, but it can be functionally defined as a decrease in the oxygen carrying capacity of the blood. That means anemia can arise if there is insufficient hemoglobin or that the hemoglobin has an impaired function. Also, anemia is a decrease in red blood cells number or function. Anemia may result whenever red blood cell production is impaired or that when the RBC lifespan is shortened or there is blood loss. The different causes of anemia would be a disorder in the iron kinetics and the heme metabolism, any defects in the DNA metabolism, bone marrow failure, an increased destruction of these red blood cells, and structural defects of hemoglobin. Anemia is caused by a defect in the iron kinetics and heme metabolism. So why is iron important? So iron is absorbed from the diet in the small intestines, and then these are carried by transferring to a cell in need, and these are brought into the cell where it is held as a ferritin or incorporated into its final molecule, which is hemoglobin. So iron functions in oxygen transport, and the three compartments that iron is stored is in ferritin, transferrin, and hemoglobin. So the iron, any loss of iron when there is inadequate stores, or that means iron that is stored in these compartments, we call that as an iron deficiency anemia. But if there is adequate stores but there is an impaired iron mobilization, like in rheumatoid arthritis, TB, HIV, or cancer, we call this now as the anemia of chronic inflammation. Sideroblastic anemia is another type of iron anemia where there is a presence of non-heme iron in developing RBC. The non-heme iron is the problem in the RBC. This is caused by chemicals, for example, lead. The hematologic tests to determine the types of anemia by iron kinetics are as follows. We can use hemoglobin, serum ferritin, serum iron, and total iron binding capacity. Hemoglobin is where about 90% of the iron stores are stored. So a decrease in the iron stores would indicate a decrease in hemoglobin. Serum ferritin would reflect the iron stores of the body tissues. Serum iron is also a sensitive indicator for iron depletion. And TIBC, or total iron binding capacity, can measure the blood's capacity to bind iron with transferrin. These are the different tests showing the results for each of the different conditions. So we have hemoglobin, ferritin, serum iron, and TIBC. For iron deficiency anemia, everything would be decreased except for TIBC. TIBC is increased. For anemia of chronic inflammation, everything will be decreased except for serum ferritin, which is an increase. And in sideroblastic anemia, everything is increased except for hemoglobin, which is decreased. The blood picture for IDA is that the red blood cells are microcytic, hypochromic, and with anisocytosis. While in anemia of chronic inflammation, the red blood cells appear normal. They are normocytic and normochromic. While in sideroblastic anemia, where the problem is there is normal iron stores, or the iron is abundant in the bone marrow. That's why ferritin, iron, and TIBC are all increased or normal. But the problem is the body is unable to use it to make a hemoglobin. That's why hemoglobin is still decreased. So again, the difference of sideroblastic anemia is that there is no problem with the storage or the amount of iron. There is an increased amount of iron available for use. It's just that the body does not know how to convert it to a proper iron in hemoglobin. Blood picture for sideroblastic anemia is that they are normocytic and normochromic. And when the bone marrow smear is stained with a Prussian blue stain, okay, ringed sideroblasts may be seen. This is what ringed sideroblast looks like. So these are normal blasts surrounded by bluish iron deposits. So this shows the presence of iron stores in the bone marrow that may be used for hemoglobin. But again, the body does not know how to use it. So the presence of ringed sideroblasts in the bone marrow is a hallmark for sideroblastic anemia. The next category is anemia that is caused by defects of DNA metabolism. An impaired deoxyribonucleic acid or DNA metabolism can cause systemic effects by impairing production of all rapidly dividing cells. And an example of this type of cells are the hematopoietic tissues, because they must be replenished throughout life, and any impairment of cell production is evident. The hallmark for this type of defect is the megaloblastic anemia. Megaloblastic anemia is a type of anemia that is caused by defects of DNA metabolism, and this is commonly caused by a folate deficiency or a vitamin B12 deficiency. Out of all of the causes leading to megaloblastic anemia, these vitamins, folate and vitamin B12 deficiency, are the common causes. These two vitamins are very important they are the building blocks, or they are important in the production, or essential for the production of healthy cells. We have to differentiate megaloblastic anemia with pernicious anemia. Although pernicious anemia is a type of megaloblastic anemia, the difference is that in pernicious anemia, the body is not able to absorb the vitamin B12 due to the lack of the intrinsic factor present in the stomach secretions. So in pernicious anemia, There is vitamin B12. Again, vitamin B12 is not deficient in pernicious anemia. It's just that the body cannot absorb the vitamin properly, also leading to anemia. There are two types of tests that can identify megaloblastic anemia. We have the screening tests and the diagnostic tests. For the screening tests, a peripheral blood smear is reviewed and the following should be seen. We have the ovalocytes, the chryocytes, macrocytes, Howell-Jolly bodies, and hypersegmented neutrophils. For the diagnostic tests or the confirmatory test, we have the bone marrow examination, which is the reference confirmatory test. But nowadays, there are other modern tests used to identify these type of disorders. In the bone marrow, what we are looking for is the presence of megaloblasts. So these are megaloblasts. They are present in the bone marrow. Megaloblasts are large RBC precursors, and these are destroyed in the marrow and are not released into the circulation. But there are still some megaloblasts who may mature to become large red cells, and this is what we call as the macrocytes. So the megaloblasts that mature as macrocytes reach the circulation. And this is what we see in the screening test. But these macrocytes do not function normally. The next category is anemia due to bone marrow failure. Bone marrow failure is the reduction or the cessation of blood cell production, which affects one or more of the cell lines. An example of a disorder under this category is aplastic anemia. In aplastic anemia, there is a bone marrow failure resulting to damaged or defective stem cells. So this is a bone marrow in aplastic anemia, and this is a bone marrow in a healthy individual. As we can see in the first picture, there is an abundance of stem cells, hematopoietic stem cells, producing new cell lines, while in aplastic anemia, there is defective or damaged stem cells. Pancytopenia means that there is a decrease in number of circulating red blood cells, white blood cells, and platelets. When all three cell lines are decreased, we can call this as pancytopenia. The different causes on why the bone marrow might fail might be the following. First, there might be a destruction of hematopoietic stem cells, so there might be something causing to destroy the stem cells, or a premature apoptosis of hematopoietic stem cells. There may also be an ineffective hematopoiesis, or maybe the bone marrow microenvironment is disrupted. There may also be a decreased production of hematopoietic growth factors and the other related hormones, or there is a loss of normal hematopoietic tissue. So these were the mentioned causes of bone marrow failure earlier. We have failure of bone marrow production, increased destruction, ineffective erythropoiesis, bone marrow infiltration, bone marrow hypoplasia, and ineffective hematopoiesis. And these are examples of certain conditions or disorders that are caused by or that results from these causes. For bone marrow production, we have aplastic anemia, as mentioned earlier. For increased destruction of blood cells, we have paroxysmal nocturnal hemoglobinuria. For ineffective hematopoiesis, examples are myelodysplastic syndrome and megaloblastic anemia as well. For bone marrow infiltration, we have acute anemia and hairy cell leukemia. The hairy cell leukemia is mentioned in the other videos. Bone marrow hypoplasia, we have Fanconi's anemia, dyskeratosis congenita, Diamond-Blackfan syndrome, and Shwachman-Bodian-Diamond syndrome. And lastly, for ineffective hematopoiesis, we have congenital dyserythropoietic anemia. As mentioned earlier, an example of anemia of bone marrow failure is aplastic anemia, and these are the characteristic features of aplastic anemia. There will be pancytopenia meaning that there will be a decreased number of circulating RBC, WBC, and platelet. Reticulocytopenia. Reticulocytopenia is the abnormal decrease of reticulocytes in the body. Bone marrow hypocellularity, meaning that there is a decrease in hematopoietic cells and there will be a depletion of hematopoietic stem cells. As seen again in this picture, this is a normal hypercellular bone marrow, while this is a hypocellular marrow with the absence of the decrease or the depleted stem cells. The last category is anemia due to the destruction of erythrocytes. This destruction of erythrocytes is known as hemolysis because hemolysis is the lysis or the rupturing of red blood cells. When RBCs rupture, the contents are released into the surrounding fluid. When hemolysis happens, the number of red blood cells would be reduced, therefore also reducing the oxygenation capacity of the blood and causing anemia. But for hemolytic anemia to happen, the rate of RBC destruction should exceed the increased rate of RBC production. Please notice here that there is an increased rate of RBC production, meaning that normally the bone marrow should be able to compensate by accelerating the production of RBC so that the RBC destroyed can be replaced. But for hemolytic anemia to happen, a significant RBC destruction must occur before it happens. So the destruction is more compared to the production. So the bone marrow is not able to compensate for the destruction. Anemia due to destruction of erythrocytes may be classified as follows: acute versus chronic, inherited versus acquired, intrinsic versus extrinsic, and intravascular versus extravascular with fragmentation versus macrophage mediated. Every hemolytic condition can be classified according to these descriptors. Acute hemolysis have a rapid onset and is isolated, sudden, episodic, or paroxysmal. Chronic hemolysis may not be evident if the bone marrow is able to compensate, but it may be punctured over time with hemolytic crisis that can cause anemia. Inherited hemolytic conditions are passed to offspring by mutant genes from the parents, while acquired hemolytic disorders develop in individuals who were previously normal but acquire an agent or condition that lyses the red blood cells. Intrinsic is caused by a defect in the red blood cells themselves and result when one or more of the genes that control red blood cell production do not function properly, and this is often inherited. So intrinsic means an RBC defect in its own. Extrinsic is caused by the action of external agents, and when we say external agents, these are conditions that arise from outside the red blood cell. Intravascular hemolysis occurs by fragmentation, and also although this takes place in the bloodstream, this can also be done in the spleen and bone marrow. Extravascular, meaning that it is outside of the blood vessels, and this can refer either to the lysis within the macrophage or not in the bloodstream, or to the fact that macrophages are in the tissues, chiefly in the spleen and the liver, and thus are outside the vasculature or outside the blood vessels. The next slide shows a table that has been lifted from Rodak's hematology book. So it shows the classification of selected hemolytic anemias by primary cause and by the type of hemolysis. So some conditions may exhibit mixed presentations under certain circumstances, but what is evident is that in hereditary conditions, so that would be hereditary membrane, enzyme defects, and globin abnormalities, lead to chronic hemolysis, and these are the ones in red letters. And then acquired conditions, and this is everything on top, would often become acute, acute hemolysis seen in green writing. Furthermore, the intrinsic defects, so that's all of these, would lead into hereditary or are caused by hereditary conditions, while extrinsic defects are or extrinsic factors lead to acquired hemolytic disorders. So how does the peripheral blood smear look like when there is anemia due to the destruction of erythrocytes? We will see the following. Spherocytes will be seen in the blood smear, elliptocytes and evalocytes. Acanthocytes will be seen, Burr cells, keratocytes, schistocytes, blister cells. The Bite cell and Heinz-Ehrlich would be specifically seen for G6PD deficiency. So this is the Bite cell and the Heinz-Ehrlich bodies. Also, RBC agglutination may also be seen. This is to note that jaundice refers to the yellow color of the skin or the sclera. Icteric is a term which describes the plasma and the tissues. An increase in plasma bilirubin would give a yellow tinge not only to the plasma but also to the body tissues. It is readily evident in the sclera of the eyes and the skin, especially in fair-skinned individuals. Hemolytic anemia would always have jaundice and any or all of the following red blood cells. And the last category is the anemia due to abnormal globin development. Hemoglobinopathy refers to a disease state involving the hemoglobin molecule. All hemoglobinopathies result from the genetic mutation in one or more of the genes that affect the hemoglobin synthesis. All hemoglobinopathies affect hemoglobin synthesis in one or two ways, either qualitatively or quantitatively. And based on this distinction, hematologists divide hemoglobinopathies into two categories. We have the first category as the structural defect hemoglobinopathy, and these are the qualitative defects. And the second category, or the second category is thalassemia, which is the quantitative defects. These are examples of structural hemoglobinopathy defects. We have sickle cell, hemoglobin S, and hemoglobin SC Harlem. For sickle cell, abnormal hemoglobin is hemoglobin S, where in the sixth position, glucose is replaced by valine. Solubility test is positive. And in the peripheral blood, we will find sickle cells. Other notable red blood cells would be target cells, nucleated RBCs, polychromasia, Howell-Jolly bodies, and basophilic stipplings. While in hemoglobin C disease, the abnormal hemoglobin present is hemoglobin C, and glucose is once again in the sixth position replaced by, but this time lysine, giving a negative solubility test. The hallmark for identification would be the hemoglobin C crystals found in the peripheral smears. Other cells seen would also be target cells, nucleated red blood cells, and occasionally some microcytes. For hemoglobin SC Harlem, we will have hemoglobin C Harlem as an abnormal hemoglobin and hemoglobin S. It has a positive solubility result, and we will find target cells in the peripheral blood. The second chart is now about thalassemia, which is the quantitative defect of the globin development. So for a normal hemoglobin, there should be 4 functional alpha globin genes. But in thalassemia, a silent carrier, there is a loss of one allele or the deletion of one alpha globin gene, leaving only 3 functional globin genes. There is no reliable way of diagnosing silent carriers as the hematologic tests are normal. There is no hematologic abnormalities. The way to diagnose silent carrier is to test for genetic analysis. The next one is thalassemia trait, where there is a deletion of 2 alpha globin genes. Thalassemia minor exists in 2 forms, the homozygous and the heterozygous. This syndrome is asymptomatic and is characterized by mild microcytic anemia. The next one is hemoglobin H, where there is a deletion of 3 alpha globin gene, and only 1 alpha gene remains to produce the alpha chains. This is characterized by mild to moderate microcytic and hypochromic anemia with poikilocytosis, including target cells and other bizarre shapes. When this is incubated, when these red blood cells are incubated with brilliant cressyl blue or new methylene blue, the RBCs would display hemoglobin H, and this is what we call as the golf ball or the raspberry cells. The last one is alpha thalassemia major, where there is a loss of 4 alleles. Anemia begins before birth, so in this state, the fetus does not survive. That's why it is also called hydrops fetalis. At delivery, there is a severe microcytic hypochromic anemia with numerous nucleated RBCs. So again, for alpha thalassemia major, there is the absence of 4 alleles and non-existing normal alpha globin genes to produce the alpha chains. This chart is now about beta thalassemia. Beta thalassemia is divided into 4 categories, which is based on the clinical manifestations. The first one is the silent carrier, where a gene mutation, which is this one, produces only a small decrease in the production of the beta chain. The silent carrier state results in no hematologic abnormalities. The second one is beta thalassemia minor, also called the beta thalassemia trait, results when one of the beta globin genes is affected by mutation, either decreased production, which is noted by the plus sign, or an abolished production, which is noted by a 0 sign, whereas the other beta globin gene is still normal. It usually presents a mild asymptomatic anemia. The red blood cells are microcytic and hypochromic with poikilocytosis. Basophilic stipplings may also be seen if right stain is used on the peripheral smear. The third beta thalassemia is beta thalassemia major, where either none, so that's 0, or a decreased amount of beta chains are produced. This is characterized by severe anemia that requires regular transfusion therapy. So again, beta thalassemia major always requires transfusion. The peripheral blood film shows marked microcytosis, hypochromia, anisocytosis, and poikilocytosis, including target cells, teardrop cells, and elliptocytes. RBC inclusions are commonly found, including basophilic stipplings, Howell-Jolly bodies, and Pappenheimer bodies. The last type is the beta thalassemia intermedia. So this is a term used to describe anemia that is more severe than minor, but does not require the regular transfusion of the major. So beta thalassemia intermedia is a condition found in between the minor and the major. So these are the patients where they fall in between the observed beta thalassemia trait, but does not need regular transfusion. Clinical manifestations can vary from minimal symptoms to severe exercise intolerance. So that ends this video about red blood cell abnormalities and red blood cell disorders. So again, this is just an overview. The specifics of the disorders were not discussed. Comment down below if you want any of the disorders to be emphasized. Also, make sure that you watch the other 2 videos about abnormalities and disorders, which is WBC and platelet abnormalities. Thank you for watching.